Hello and welcome to Panome Bio's discussion today entitled From Payload to Patient, Real-World Applications of Metabolomics and ADCs and Immuno-Oncology. My name is Chris Eden. I'm a Senior Director at Panome Bio. I have with me today Dr. Gary Patti, Panome's CSO and co-founder, as well as the person who established the next-gen metabolomics platform that we use. Gary, welcome.
Thank you. Thank you very much. Pleasure to be here.
Yeah, and today I just have, four common questions that I get from pharmaceutical scientists that maybe, we could go over. The first question I have is, a lot of focus as of late is on ADCs for cancer therapies. And how can untargeted metabolomics help with optimization of ADCs?
And maybe just for the people watching that may not be familiar with ADCs, you could define what you mean by ADCs.
Oh, sorry, yeah.
Yeah.
ADCs are antibody drug conjugates.
Yeah. So it's a good question. I think, and we'll talk a little bit about how metabolomics could be applicable to the specific question. I think that what I'm going to say here is actually broader than that, beyond these particular class of this particular inquiry that you're referring to. That actually a lot of drugs could fall into the benefits here, and that is. How do you know if your, in this case, antibody conjugates or whatever it is that you're thinking about are working? It could be a drug in general or this particular type of drug. And historically and classically, we tend to rely on phenotype. How are the people doing? With the ultimate endpoint being death. And that's useful, of course, because that is the readout that we care about. We want to get better and not die. That's how we want these drugs to work, but it does take a while to do. And there's a lot of costs associated with that. The other concern that you have with these drugs or any kind of chemo agent is the toxicity. The idea here is that you're introducing drugs tethered to antibodies because you're trying to target them specifically to a particular cell type, cancer cells. And if you don't target the cancer cell, the reason you do that is because these drugs, have toxicity profiles associated with other cell types. That's why you try to conjugate them so that you can specifically deliver them to one cell type. That's one of the challenging things about cancer or any disease cell type in general. It's easy usually to come up with ways to kill that cell type. But it's tricky to come up with a way, like, for example, you could put cancer cells in addition, dump bleach on them, that's going to kill them. But if you expose somebody to bleach, you're going to kill all the cells in the body. So the real thing is how specific is this? And if it's not specific, what does that mean? It usually means toxicity. And so how toxic is something and how long do you have to do a trial or how long do you have to administer these sorts of compounds before you can get a sense for whether something is toxic or not? And I think one area where metabolomics has a lot of potential is to get to that message a little faster. So instead of trying to look for an endpoint of death or looking for severe toxicological profiles that are associated with organ failure or whatever it may be, can you find markers, circulating markers that indicate the drugs are working well against the tumors? It's indications that the drugs actually are eliminating or improving at least decreasing tumor progression. Can you find those sorts of circulating markers? And furthermore, are there markers that are signatures of toxicity that you can do, that you can profile? The idea being there is if you could give your conjugate, your antibody conjugates, and then for a short period of time, say 24 hours, and then do a blood test later, and you could find out if they're working and if there's any off-target effects. That would be a lot more efficient than having you do experiments or trials that last significantly longer than that. Particularly if the trial isn't going to be successful. If the trial is not going to be successful, you want to know as fast as possible.
No, that makes sense. No, thank you. No, that's great. The other question that I commonly get is, it's a common term. In immuno-oncology, they use the term for a tumor as being hot or cold in responsiveness to immunotherapy or not. Have you seen similar metabolic signatures for hot or cold tumors that could be used for stratification purposes?
Yeah, I mean, I think tumor stratification at the metabolic level is unquestionably informative. I mean, the phenotype, the metabolism of a tumor in many ways is synonymous with its phenotype. Certain metabolic processes are conserved largely among a lot of different tumors. The classical example being the Warburg effect. We know that cancer cells consume a lot of glucose, and that a large stoichiometric fraction of that glucose is converted to lactate and excreted. So that's a phenotype that's conserved, but there are many other different kinds of metabolic phenotypes that may or may not be present in different kinds of tumors. And even the Warburg effect, if you take a whole panel of cancer cells and you look at how much glucose they uptake and how much is excreted as lactate, you see a lot of variation in that they're not all equal. And so the metabolism of the tumor is a reflection of what it is doing biochemically and how it is processing nutrients and what pathways they're using. And so I think it's actually very compelling to imagine not only, there's a possibility of targeting the metabolism. That's often what we think about when you think about metabolism, like antifolates. Can you identify a pathway in cancer cells that's active that you can go after and target therapeutically? But you can also think of it as a way to stratify your populations of tumors. So yes, you have, melanoma, but there's a lot of different kinds of variations of melanoma, and even the same oncogene can drive different types of phenotypes. And those phenotypes might be not just driven by other genetics factors, but it's also influenced by environmental factors, diet, exercise, just the environment as a whole. And so I think all of those, and that's a really interesting one as well, actually. Like we're learning, diet is a great example. What diet people consume does have an impact on how well drugs work. There's many instances of that now, and it's very likely that certain diets will augment different kinds of therapies and maybe make them work better or maybe make them work a little less. And so trying to figure out what's happening there, not just at the phenotypic level of trying to stratify the tumor, but trying to understand the person, because tumors don't happen in isolation. They're connected to the physiology of the entire patient. And trying to understand that stratification as well. This person is in this environment or has this other physiological background, and therefore this drug will work or won't work, I think will be a key piece to it. There's a lot to do there, of course, a lot to discover, but I think the evidence is pretty strong that there are correlations among all those things.
That makes sense. Thank you. The question 3 is, and you kind of answered this, you kind of alluded to this anyway, is where do you see metabolomics fitting into the immuno-oncology? It's a very general question.
Yeah, no, I mean, immuno-oncology, the connection between immune cells and cancer cells is very intimate, the relationship between them. And cancer cells do a lot of it's well established that cancer cells can reprogram cells in their surrounding. Immune cells can also influence the metabolism of cancer cells. I mean, they're tightly interconnected through metabolism. And one example, there's 2 ways that cells that are in the same environment in a tumor, say, or sometimes we use the word microenvironment, the way that those cells interact, whether it be immune cells and cancer cells, or actually you could extrapolate this to really any kind of stromal cell that's present in the tumor. And that is, they can interact competitively or they can interact in a cooperative way. And so let me just take a minute to tease that apart. So what I mean by competitively is that if and there's examples of this already between immune cells and cancer cells, but if one cell type consumes all of a given nutrient, and it has a much higher avidity for that given nutrient, it can actually prevent the other cell type from accessing that nutrient. And in some cases, some beautiful work out of Erica Pierce's lab that showed some of the seminal work in this space, that basically boxing that cell type out of that nutrient can be then detrimental to that particular cell type's fate. So that's one way, that's the competitive phenotype. But cancer cells are smart, and they also have the ability to hijack, sometimes we use the word activate, other cell types in their environment. So they can take cells and they can co-opt their metabolism and rearrange it such that that particular cell type starts producing a nutrient that gets output and then utilized by the tumor, by the cancer cells specifically. And so they have this sort of symbiotic relationship where the cancer cells say, I really need nutrient X. I'm lazy. I don't want to make it. I'm going to rely on that other cell type over there to make nutrient X and cargo it to me so that I can depend upon that. So I think those two types of relationships are reflective of the interactiveness and the symbiotic nature in which immune cells and cancer cells, and in fact, all other stromal cells in the microenvironment interact.
That makes sense. I think there's maybe you refer to it, there's a CAR T paper that shows that the CAR T cells the tumor would take up all the cholesterol of the CAR Ts and you kind of starving out the CAR Ts and
Exactly.
um,
Yeah. Yeah. And there's examples like that with glutamine, glucose, there's a lot of nitrogen, several, those types of examples are prominent.
very cool. Um, and the last question I have is, it's a very broad question again. It's why are groups are hesitant to use metabolomics as a standard method for biomarker development in your opinion?
Yeah, I mean that's a tricky question. To try to assess why someone is or isn't hesitant. Um, Metabolomics is the newest kid on the block for these omics assays, and so it often takes time for organizations to assimilate, get comfort levels, and so forth. A lot has happened in metabolomics as well. I think some organizations that with whom I've interacted they maybe tried metabolomics 10, 15 years ago, and maybe their experiences weren't, they didn't extract the kind of insights they'd hoped and have therefore not revisited it. And I guess maybe that the message for that particular individual is that metabolomics has come an extraordinary way over the last 10 or 15 years. The technologies have undergone just amazing advances, both on the hardware side, and perhaps even more importantly, on the software side, where we're now able to do quantitation much better and to get much more robust, reliable results that enable us to find changes that are much less striking. It doesn't have to be this smoking gun, major dramatic change in a compound to find it between sample groups, because we've been able to figure out how to handle the noise and improve the fidelity of the data. We're now much better at finding even small little biochemical differences between sample groups. And so, I think as more and more success stories with metabolomics roll out, that it provides increasing numbers of precedents for the utility of metabolomics, and hopefully will be exemplary for some of these other folks who maybe tried metabolomics or in the earlier days of the technology, that they might come back to it and see how far it's come.
No, I appreciate it, Gary. That makes a lot of sense.
Yep.
But I appreciate your time today. Those are all the questions I had. Thank you again for attending this.
Yeah, absolutely. My pleasure. Thanks, Chris.